To begin, let's talk about how a micropipette works, because we will be using one in today's demonstration. A micropipettor allows you to accurately withdraw and dispense small volumes of liquids, usually less than 1 milliliter. The design can be quite complicated, but air displacement pipettes like this one all operate on the same principle. A piston is positioned in a cylinder so that when it is moved, it creates a vacuum. This vacuum is used to fill a pipette tip with the desired volume of liquid. Opposite movement of the piston allows the liquid to be expelled where needed. In the diagram on the right, look at number 1. The piston moves to the appropriate position when the volume is set. This is usually done by turning an adjustment knob found on the handle. In number 2, we see that when the operating button is pressed to the first stop, the piston expels the same volume of air as indicated on the volume setting. In number 3, after immersing the tip into the liquid, the operating button is released. This creates a partial vacuum and the specified volume of liquid is aspirated into the tip. Number 4. When the operating button is pressed to the first stop again, the air dispenses the liquid. To empty the tip completely, the operating button is pressed to the second stop, also known as the blowout.
Welcome to the pipetting tutorial How to Pipette Correctly. I'm using a 2-button pipette. It has a control button with 2 stops and an ejector button. Pipetting starts when you attach the tip. Apply light pressure to insert the pipette into the tip. There is no need for force or moving from side to side. This would only strain your arm and damage the pipette. When aspirating liquid, 2 factors influence accurate liquid uptake: the aspiration angle and the immersion depth. The aspiration angle should be vertical. Otherwise, too much liquid is aspirated. The immersion depth, meaning the depth of the pipette tip inside the liquid, should be as large as necessary but as small as possible. If the tip is immersed too deeply, additional liquid can be easily aspirated due to the capillary effect. Press the control button down to the first stop and immerse the tip into the liquid. Once the aspiration angle and immersion depth have been checked, slowly release the control button. Press the control button down to the first stop again and aspirate once more. Repeat this 2 to 3 times to saturate the air in the tip. When dispensing liquid, the angle of the pipette should be between 20 and 45 degrees, whilst the tip should stay in contact with the vessel wall. This guarantees optimal flow out of the liquid into the target vessel. Press the control button down to the first stop. Then perform the blowout by pressing further down to the second stop. This ensures that the tip is emptied completely. The last step during pipetting is ejection of the tip into a garbage bin by pressing the ejector button. Achieve accurate and reliable pipetting results with soft tip attachment, vertical aspiration, a low immersion depth, and a 20 to 45 degree dispensing angle directly in contact with the vessel wall. So, improve your pipetting skills.
Today we are going to be looking at our first method of isolating bacteria, called pore plating. In order to study a particular bacterial species, you must first make sure that you have a pure culture of it, and that begins with isolation. In this simplified diagram, we see that we mix a bacterial suspension with something called molten agar, and then after we allow it to incubate, we see colonies growing on the surface and within the agar. We could then harvest one of those colonies and establish a new culture which we could then consider to be a pure culture. Here we see a tube of contaminated Bacillus thuringiensis culture that we will use in the pour plate procedure. First, we will need an empty sterile petri dish, labeled correctly as shown. You can see the initials, date, and a description of what will be in that plate. Today we will be using a micropipettor that has a working range of 20 to 200 microliters. Micropipettors are commonly referred to as having a size, which is almost always the upper limit of its range. So we would commonly call this a 200 microliter pipettor. We first need to adjust it so that it will dispense 100 microliters by rotating the adjustment knob until we see 100 in the window. You should never try to adjust a micropipettor beyond its working range. Each micropipette size uses a different size disposable tip. Make sure the tip size you are using is the right one for your pipettor. It is a good idea to seat the tip firmly and correctly onto the handle by tapping it a few times to make sure that the tip does not fall off or leak air. Withdraw 100 microliters of the contaminated broth culture and then dispense that into the bottom of a sterile empty petri dish. And then dispense of the contaminated tip into an appropriate receptacle. You will then need to get a container of a sterile nutrient broth that has a solidifying agent mixed into it called agar. This is commonly used to create a gel-like solid medium in bacteriology because it is non-nutritive for almost all bacteria. Also, its melting characteristics are desirable for making solid media with ingredients that might be heat sensitive. Agar melts at about 85 degrees Celsius, but once melted it does not completely re-solidify until it reaches about 35 to 45 degrees Celsius. When the molten agar has been cooled to below 60 Celsius, it can be safely poured into the dish with the culture without killing a significant number of cells by heat. Once poured, the agar culture mixture should be mixed gently but thoroughly by swirling as demonstrated here. Switch swirling directions once or twice to aid in complete mixing. Here, I will remove the lid so that you can see the desired level of swirling. You want to swirl the mixture enough so that you create a visible wavefront of liquid, as seen here, but not without doing it so strongly that it splashes up and over the rim of the plate. If that happens, the plate cannot be used and should be discarded because you cannot be sure if it is not contaminated with any other organisms accidentally introduced into the mix. The end result may look something like this, depending on the concentration of cells in the original sample. This plate shows individual colonies that are each derived from one single cell, but they are growing so close to one another that some colony overlap is possible, and that it would be very difficult to try to remove one colony from that plate without touching another. In this case, you would repeat the procedure with a more dilute sample.